for the 7th step of molecular docking course. We are going to predict ADMET properties of ligands and drugs. For this purpose, we will go to SwissADME as well as admetSAR website. So first we will go to SwissADME site. So we will open the browser. We will type the website of SwissADME, and for this prediction of ADMET values, we have to create the files in an Excel format. So we created the 4 files from the ligands smiles as well as the drug smiles. Okay, so the last 2 molecules are considered as the drugs, the other 4 molecules are the ligands. So we will copy down all the 6 smile forms and we will paste it in a window, and then we will click the run button. Now we will get the predictions, SwissADME predictions of the ligands and drugs. So if you see, this is the radar prediction which represents 5 properties of the ligands, which clearly indicates that if the ligand is present in the pink region, then it's considered to be as a good drug molecule. So if you see this, first ligand is well within that group, and second ligand is also well within that region, third is also well within the region, and fourth is also within the region, even though it is found to be a little bit in the extreme region, but within the pink color region it is present. So the selected 4 molecules for our present study is found to be good for drug likeliness, and with the selected drugs, that is the 5th and 6th, so they show that out of range. See, so our hits might be better a drug molecule than the already available drugs as per this ADME predictions from SwissADME. For example, if I want to highlight one point, you can see here Lipinski rule. You can see this, it will have a 0 violation, and this compound is also have a 0 violation, and this compound is also have a 0 violation, and this compound is also have 0 violation. It clearly shows that this compound is better drug-like molecule. Similarly, the bioavailability score will be 0.55. This will be 0.55, and this is also 0.55, and this is also 0.5. So almost all the molecules will have same bioavailability score according to ADME. So according to the SwissADME result, either of these 4 molecules are found to be better to be considered as a drug molecule. And we can also see the boiled egg image. It also clearly indicates that the molecule 1 and 4 are found to be hydrophilic in nature, and 2 and 3 are found to be hydrophobic in nature. So depending upon the requirement, we can take any one of this molecule. Okay, so this is what the outcome of SwissADME result. If you need this result in a text format, then you will save this retrieved data. So as a CSV format, you can download the SwissADME results. Okay, so with the SwissADME, we can choose all the 4 hit molecule is a better molecule. Then we go to the next server, that is admetSAR. Okay, so for that admetSAR prediction for ADMET properties. So we'll go to this admetSAR. It's better to use Chrome for the admetSAR prediction. So we open Chrome, and we can submit a text file for this admetSAR prediction. So we will use this ADMET screening option, so that we can submit our text file by clicking this place. We can submit the smiles text, which is already created with the smiles. Here you can see this smiles text. So all the smiles are pasted one by one, and this smile file is actually uploaded here, and then we have to click this submit button for the prediction of admetOx properties through this admetSAR server. Once the predictions are completed, you can get the results through this admetSAR input link. So this admetSAR is also going to predict the properties, whether the molecules will be act as a drug or not, metabolic properties, distribution properties, toxicities, everything can be predicted with the help of this particular server. So we have to refresh this point. Once results are ready, it will be displayed in this page. Now you can see the results. So the first compound, again you will have a radar structure, second compound, third compound, fourth compound, and this is drug, and this is also drug. So again this is also shows that the drugs will have a values out in the outer range. This drug is also having a outer range, but our compound will have these values are inner range of the allowed region, that is lower drug like upper domain and drug like lower domain. So in between this our compounds are present. So that is what it indicates. So our compounds are better than the drugs. It's also proved by this admetSAR prediction. If you want to save this file, you can click this text option to save this file as a text format. And then if you want to see the details of each compound, you click this view button, and then you will see the further predictions of individual headings like basic properties of this particular molecule, like hydrogen bond donor, hydrogen bond acceptor, total polar surface area, and log p value. So number of atoms which are available, molecular weight. Like this we will see the properties. See here Lipinski rule, it accept 100% of Lipinski rule is accepted here, and you can say Pfizer rule, GSK rule, everything is accepted for this compound. And then the metabolism, whether it is going to, how it will affect the metabolism, how it will affect the excretion, how it is distributed. So everything is given in this particular page. You can see this, whether it is going to inhibit the metabolizing enzyme. See, very less amount of percentage is inhibiting this enzyme. So like this, further properties of the compounds, admetOx property, whether it is going to be toxic to human, which organ it is to human, which animal it is toxic. So like this, you can see nephrotoxicity, eye erosion. So what is the percentage of its toxicity can be predicted here. Even the how much endocrine system will be affected by this particular molecule is also predicted here. And then you can see the ecological, environmental toxicity of this particular molecule is also predicted. And then you can cosmetic toxicity, if you want to use this chemical as a toxic cosmetic agent, whether it will affect the eye, skin, that is also predicted here. So like this, individual compound prediction also available in this admetSAR. So you can save this information of all the compounds in the CSV format. So like this, you can save all the results of this admetSAR individually as a whole. So by knowing all this admetSAR result as well as the ADME results, we can find out which is the best compound. So from these two results, we came to know that all the hits are found to be better than the drug molecule according to the admetOx prediction.